Greetings to all. I am Dr. Poopathi, Senior Scientist at the cadre of Scientist G in Vector Control Research Center under the umbrella of Indian Council of Medical Research at Pondicherry. First of all, I would like to convey my sincere thanks to the President of Society for Vector Ecology and my Director Dr. Ashwini Kumar for providing this excellent opportunity to spare some of the information about the Microbial Bio-pesticides for mosquito control program. As we all know, mosquitoes are great nuisance causing dangerous diseases such as malaria, filaria, dengue, chikungunya, Japanese encephalitis, etc. These diseases pose major problem in disease prevalence countries. Mosquitoes are flies coming under the family Culicidae. Over 300 species of mosquitoes are known to exist in the world. Mosquitoes can be found in their variable habitats at the breeding sites. Because of their blood-sucking habitats, mosquitoes are prone to transmit disease-causing organisms such as viruses, for example, dengue, equine encephalitis, protozoans like malaria, filaria, nematodes like that. Bancrofting filariasis from the vertebrate host to the human and the animals also. The importance of mosquitoes in human and the animal diseases have made them an important target of medical, veterinary, and the conservation research. Mosquitoes are typically good source of the food for organisms such as larvae, larval eating fish like gambusia, immature dragonflies, damselflies, aquatic beetles, etc. Adult mosquitoes are also the source of the feed materials for the terrestrial animals, for example, the frogs, lizards, birds, etc. Mosquito larvae are the filter feeders, but it is not clear exactly what type of the microorganisms they prefer and what mechanisms they exactly involved in these ones. So coming to the mosquito control program, there are two major strategies for vector control. That is the insecticide-treated bed nets and the indoor residual spraying. The insecticide-treated bed nets is the preventive control measures as it involves protecting the individuals with bed nets as most mosquito bites occurs at night. Indoor residual spraying involves the use of insecticides which results in the death of mosquito as they reset after feeding. Mosquito control is divided into three types: physical control, biological control, and the chemical control. Physical control is to eliminate the number and size of the frequency of mosquito population. And chemical control involves the use of chemicals designed specifically for control of the immatures and the adults. Biological mosquito control is the use of other mosquito predators. See, synthetic insecticides have been effectively used during the past several decades for mosquito control operations. And there are several synthetic insecticides, for example, malathion, chlorpyrifos, pyrethrin, cypermethrin, permethrin, etc. All these synthetic insecticides are used for control of the adult mosquitoes, but there are certain limitations, especially for the development of the resistance. So, whatever these synthetic insecticides were spraying in the field, the adult mosquitoes developed the resistance. So, other than this resistance, there are some other problem. For example, the environmental pollution, bioamplification is harmful to non-target organisms. So, the development of the insecticidal resistance, environmental pollution, bioamplifications of food chain and harmful effects on beneficial insects have led to the search for new microbial biolarvicides, especially the bacteria-based biolarvicides. So, globally, there are two bacteria, so as on date I identified, more than 2 decades before that bacteria isolated, that is, one is for the Bacillus sphaericus and other is for the Bacillus thuringiensis and israeliensis. So, these bacteria are unique and it shows the effect against the mosquito larvae. Because of the presence of the toxins, for example, the Bacillus sphaericus has two toxins, 51 and 42 kDa proteins, that is called the binary toxins, and BTI contains the multiple toxins 134, 128, 72 and 27 kDa proteins. These multiple toxins are very effective against the mosquito larvae. So, in general, when the main advantages of this application of the biolarvicide is it is a non-toxic, non-pathogenic to wildlife, narrow host spectrum, low harmful effect to human beings, no risk to non-target organisms, and no pollution control. So, these are all the advantages for these ones. So how the toxins shows the effect against the mosquito larvae? It's another question. You see, as soon as the bacteria get involved on the system of mosquito larvae, the toxins which present the bacteria are solubilized because of the involvement of gut proteases. After this, the inactive proteins are converted into active toxins. Protoxins is converted into active toxins. For example, 51 and 42 kDa proteins is converted into 43 and 39 kDa proteins. These activated toxins is ready to bind on the seeds of receptor sites and the midgut brush border membrane. So once this activated toxins binds on the seeds of the midgut brush border membrane, it gets internalized, then leads to lysis of the cells. Finally, death of the larva occurs. This is the putative mechanism of mode of actions of the bacteria with respect to the mosquito larvae. And even when these Bacillus sphaericus or BTIs has several advantages, there are certain disadvantages also there, mainly because of the chronic applications of Bacillus sphaericus in the field condition. The mosquito larvae developed the resistance. The resistance, for example, more than 2 lakhs fold of resistance was reported globally, mainly because of the inactivations of the receptor sites. So whatever the toxin moieties binds on the receptor sites, it does not show any effect. So the toxins are released along with the food materials that is excreted from the mosquito larvae. That is why the mosquitoes flourishing very happily on the aquatic systems. So I studied one resistant protein that is called 85 kDa protein is a responsible factor which shows the resistance against the mosquito bacteria. So whatever we are applying the Bacillus sphaericus and BTI, there is a timely requirement to identify, to search for an alternative biopesticides, alternative mosquitocide in the environment. So under these conditions, our research team have extensively studied. One such study is we have collected the marine fishes from various sources, and the midgut was carefully dissected out. And in that, one particular fish that is called Luxanus sanguinis, that Luxanus sanguinis midgut was dissected, where there is one bacteria was isolated that's called Bacillus cereus, BCRC B540. This particular bacteria is very effective, and the effectiveness is due to presence of the surface layer proteins with 51 kDa proteins, that is a responsible factor. And also we have collected the remnants from the marine boards, the side walls of the marine side. The remnants are deposited. Those remnants were collected, and we have identified the bacteria. Is there any special bacteria is there or not? And we studied BTI. Is very effective, and this BTI has a very special characters. It not only shows the killing effect, but also showing the biofilm formation capacity. So biofilm formation is nowadays very important in industrial technology. So this BTI has special characteristics showing the dual functions. And this picture shows the sources of the mosquito collected from various places. For example, oil degrading soil from the marine sources, freshwater fishes, desiccated fruits, then vermicomposting. All these sources we have studied, collected, and we have identified the bacteria. And when coming to the microbiological aspects of this bacteria, some of them are the gram-negative bacteria we have isolated, and some of them are the gram-positive bacteria. And generally, the gram-positive bacteria, especially the rod-shaped bacteria, showing the killing effect against the mosquito larvae. So the methodology is a very long process, especially on microbiological process. As soon as the samples from environments are bring back to our laboratory, they are isolated by using culture medium, inoculating the culture medium, allowed to grow for about nearly 48 hours or 72 hours, separate the cell mass and the supernatant, and the cell mass goes to rigorous lyophilizations, conducted preliminary bioassays for confirmation for toxicity assay. Then after that, we will study the whole genome sequence of this particular bacteria. Then the extracellular proteins from the cell culture supernatant, that is a cell-free medium, are also studied, and we found that the proteins of that extracellular proteins also showing the toxic effects. Very recently, we have reported that these extracellular toxins from the cell-free culture medium also showing the adult mosquito attractants. So that study is also going on. So based upon all these background studies, from our research center of Vector Control Research Center under the umbrella of ICMR, we have isolated more than 30 bacterial strains. These are all the list of the bacterial strains, and this was collected from the various places, from environmental sources, for example, from the very remote places of villages around in and around the Puducherry, and bad excreta, marine fishes, etc. 30 strains we have isolated. Each strain has its own uniqueness and showing the effect against the mosquito larvae. Very upon this background knowledge, now we are going to take up one particular project to because India is a wide natural resources. We have wide type of the soils. More than 7 type of soils, major soil types that we found. So initially we are going to visit the southern regions of Tamil Nadu and Puducherry, collect the different soil types, are going to isolate the mosquito bacteria. In that, if only one bacteria is effective, it will be very useful for mosquito control measures. So based upon this particular information, I would like to finally give a very small information to all readers and scientists, researchers, and all people. India we have a wide natural resources, and we have to utilize these natural resources to identify the very potential, highly effective mosquito-cell bacteria, which will be very useful for controlling the immature stages of the mosquito, so that we will live in a mosquito disease-free environment. Thank you.